Hello students, welcome back to the channel. In this video, we are going to give you a quick revision of this chapter. I hope this video will be helpful for you. Now let us start.
Now we are starting with the eukaryotic cell. The eukaryotic cell include all protista because protista kingdom include eukaryotes unicellular, kingdom Plantae, kingdom Animalia and kingdom Fungus. There is extensive compartmentalization of cytoplasm, means we are making different compartment on the cytoplasm. How? By the presence of membrane bound organelles. They are having well-developed nucleus with nuclear envelope. And they are having the cytoskeleton also, they are having the locomotory structure also. All eukaryotic cells are not similar because we know plants and animal cells are different from each other. How? Plant cell will have cell wall, animal cell will not have. Plant cell will have plastids, animal cell will not have. Plant cells are having large central vacuole, which animal cell will not have. So these are the three things which the plant cell will have, but animal cell will not have. Now, if you see for the animals will have centriole, which the plants will not have. Is it clear? So this is the difference between the plant cell and animal cell. Please learn this diagram. From the diagram, they are asking the questions also. Please learn it. Clear? Now let us see cell membrane. It is made up of phospholipid, protein. These are the main. These two are main. But carbohydrate is also present because we know very well in case of the blood group, antigens are nothing. They are sugar only, sugar polymers. They are having cholesterol for the stabilization of the cell membrane. Now phospholipid, if you know, what is phospholipid made up of? Phospholipid is made up of glycerol, fatty acids, phosphoric acid, and choline. Why I'm writing choline? Because we are using which type of phospholipid to make cell membrane? Lecithin. We'll be studying in biomolecule. I'm just drawing the diagram. Try to understand. This is head. Head is having choline. It is having phosphoric acid. I'm writing in short. It is having glycerol and two long known as fatty acid, tail. So these are the fatty acid which is working as a tail. So the head is hydrophilic. Head is hydrophilic, also known as polar molecule, and tail is hydrophobic, means non-polar. Because of this, it is having both nature, polar and non-polar. You call it as phospholipid as they are amphipathic molecule. If you take a beaker in which you are putting the oil, here putting the water, what will be the arrangement of the phospholipid? They will be arranged this way, head facing to the water, not tail. But if in the beaker, if you are putting the oil, you are finding the arrangement is tail is facing the oil, not the head. Now, if you see the cell, inside the cell, you are finding the water is present. Cytoplasm is a fluid-filled matrix, and outside it is present, you know very well, tissue fluid. Then how the arrangement is done? They are doing the arrangement in such a way that they are protecting their hydrophobic region by making as a bilayer. So cell membrane is coming as a phospholipid bilayer to protect their tail from the aqueous environment. To protect the tail from the aqueous environment. So let us draw the diagram of the cell membrane. When we draw the cell membrane diagram, we draw it this way. This is the one layer. Right now I'm drawing it monolayer. Another layer, then we will say this is phospholipid bilayer. Both sides it is hydrophilic is present just to protect itself from the aqueous environment. On the surface, you are finding, depending upon the ease of extraction, you are finding protein. This is peripheral protein, which can be easily removed. We say peripheral protein, also known as extrinsic protein. Few proteins are embedded inside. They are partially embedded, not fully embedded, partially embedded. This is known as intrinsic protein, which I have drawn right now as a partially embedded. This is your intrinsic protein. Now, some intrinsic protein are there this way, they are totally embedded in this way, spanning the cell membrane. They are going through and through the cell membrane, making a tunnel, or you can say channel. So these are known as transmembrane or channel proteins. So this is your transmembrane or channel protein. Now let us see the NCERT. First of all, detailed structure of membrane was studied only after the discovery of electron microscope. Again question, electron microscope or light microscope or compound microscope. In which year? 1950. Chemical studies, when you are studying at the chemical level, especially on the RBC, you are finding it is actually made up of what? It is mainly composed of lipid and protein. Which lipid? Major lipids are phospholipid. Is it clear? And how are they arranged? They are arranged as bilayer, with the polar head towards the outside and hydrophobic tail towards the inner part. Why they are doing this arrangement? Just to ensure that the non-polar tail of saturated hydrocarbon, tail is made up of saturated hydrocarbon, is protected from the aqueous environment. That is why they are coming as bilayer. Okay, another type of the lipid is there, they contain cholesterol. In addition to phospholipid, they also contain cholesterol. Now see in the diagram, see here, you can see this yellow color dot as a cholesterol. Cholesterol is only working as a buffer. If suppose the temperature is increasing, membrane fluidity will increase. Then it will be very difficult to control what thing can enter or what thing will not. So easily things can enter. So cholesterol is preventing it from making it too fluid. And if it is temperature is low, chances are it can become solidified. The cholesterol is not allowing it to become solidified because its orientation is in such a way that it does not allow close packing. Is it clear? Now see, cell membrane also revealed that the cell membrane contains protein and carbohydrate. So I think so we have studied how many things with that. We have studied phospholipid it contains phospholipid, it contains steroids, which is steroid, that is cholesterol, sterols, it contains protein and it contains carbohydrate. The ratio of protein to lipid will vary depending upon different cell type. For example, in case of human RBC, we are finding protein is more, lipid is less. It can change. Sometimes protein can go to 80% also. Now, depending upon the ease of extraction, membrane protein are classified in two categories, integral protein and peripheral protein. Integral means which are embedded and peripheral means which are present easily on the surface, easily they can remove. The peripheral protein lie on the surface, integrals are either partially or totally embedded. This only I tried to explain you. So this diagram, if you see, this is sugar coming out. This is peripheral protein attached only on the outside. Is it clear? And this is sugar which is coming out. The sugar is actually helping in the cell recognition. How the cell, RBC cell knows that this is foreign cell. When RBC is given, why we do the blood matching, blood group matching? Because antibodies will identify it as foreign. How they will identify? The sugar only. So they help in the cell recognition. Clear, beta? The fluid mosaic model. Now this model was given by Singer and Nicholson in 1972. So quasi-fluid nature of lipid. Who is having the fluid nature? Lipid. Lipid is having fluid nature. It enables lateral movement of protein. Lateral movement of protein. For this, I'll give you one imagination. Do one thing. Imagine one bowl of gulab jamun. This much big bowl of gulab jamun. And these are your gulab jamun. They are floating on the surface. This is the surface. Okay, surface and your gulab jamuns are here and here. Half inside, half outside. And I think so gulab jamun will move from here to here. This can move from here to here. And this movement is very easy if the syrup is fresh right now, very fresh. Gulab jamun will move easily here to there. So lateral movement will help you to identify the fluid nature. This ability to move within the membrane is measured as its fluidity. How much lateral movement they are allowing it to go, that will help you to know its fluidity. Is it clear? And fluidity is important. Why do you want? So that you can check cell growth. You know very well, cell growth will be because of fluid nature. That's why cell extension, morphology we studied. Cell wall capable of extension. To make intercellular junctions, tight junctions, gap junctions, for secretion, exocytosis, endocytosis, or cell division, we want the fluid nature because of this only. Is it clear? Important function of the cell membrane that molecule have to pass. So you know very well, two types of transport are there, passive and active transport. Many molecules can move briefly without any expenditure of energy. We say passive transport. Now, who can do passive transport? Neutral, which are not charged. They can cross by simple diffusion along the concentration gradient. Water also goes by osmosis, you know, by diffusion only. But polar molecules, you know, polar molecules are charged particles. They cannot pass. So for this, you require carrier protein. That is why in the respiration chapter or in the photosynthesis, F0F1 particle, that is channel protein. Their diffusions are facilitated. You have to take help of carrier protein to facilitate their transport. Few ions or molecules are transported across against the concentration gradient. If it is against, that time you have to use energy. Is it clear? You have to use energy. For example, we studied in the nervous system, sodium-potassium exchange pump. So I think so up till here your concept is clear, beta. Cell wall is non-living. Okay, it is an outer covering seen in the fungus and the plants. And you know very well, cell wall is freely permeable. We studied in the transport chapter. It is freely permeable. Cell membrane is selective permeable. Cell wall is protecting the cell from mechanical damage and infection. It also helps in cell-to-cell interaction. Please understand one question from here, and they can ask you here. If you see, it is helping in cell membrane is helping in intracellular junction. Let me rub it so that you can get it idea clear. Because when they frame the question, the student will never think this way. See, beta, cell-to-cell interaction is because of the cell wall. But intracellular junction is because of cell membrane. Clear? Please remember it. The cell-to-cell interaction is because of the cell wall. Why I'm focusing? Because our mind never gives the idea that it can be cell wall. We always think it is a cell membrane. Now, algae is having cell wall made up of cellulose, galactan, mannan, and mineral. Which mineral? Calcium carbonate. Again, one question. Which mineral is present in the algae cell wall? Calcium, magnesium, potassium, sulfur. They can confuse you. So, which mineral? Only calcium. Plants are having cellulose, hemicellulose, pectin, and protein. You know very well, primary cell wall is capable of growth. That is why meristematic cells are going for elongation. The cell wall of the young plant cell, especially primary cell wall, is capable of growth. Okay, so which type of growth you can write down? That is known as intusception growth. Intusception growth means addition of the material within the existing one. This type of growth is the growth in which you are adding the material within the existing one. Only one cell wall is primary cell wall. Secondary cell wall is many layered, which gradually diminish as the cell mature and the secondary wall is formed in the inner side of the primary. And I think so, if you remember, we have already studied that primary is outermost and secondary is made inner one. Now, you can do one thing again. If you want, you can make this way. This is primary cell wall and make 2-3 lines of the secondary cell wall. This is secondary S1, secondary S1, S2, and S3. Every time it will be innermost will be fresh one. This is S1, this is S2, and this is S3. Innermost is the one newly formed secondary cell wall is the innermost, not outermost. It will be the innermost. Is it clear? Middle lamella will be seen in the plants, which is working as a cementing material. It is having calcium pectate. It is working like a glue, cementing material. So middle lamella is equal to adhering junction in the animal cell. Just like animal cells are held together by the adhering junction, which is helping in gluing. If you remember in the structural organization, same middle lamella is for functioning in the plant cell. Cell wall and middle lamella may be traversed by the plasmodesmata, which connect the cytoplasm. And I think so. I explain you plasmodesmata connection or not? Pit formation and all this thing I explained now. In that plasmodesmata connection also I explained it. Now, cell wall and cell membrane, two topics are covered. Now we have to start with the endomembrane system. But before endomembrane system, let me explain you nucleus, so that the concept get clear why endomembrane system is made, how it is made, and why it is absent in the prokaryote. See, this is your nuclear membrane. This is your nuclear pore. So this is your nuclear pore. Inside this, you are finding a network is present. This is known as chromatin. So this thread-like structure is known as chromatin. And you are finding one dense body is present here, which is spherical, not bounded by any membrane. This is known as nucleus. So let me first explain you nucleus, then we will come to the endomembrane system. So nucleus is a cell organelle which was first described by Robert Brown in 1831. Nucleus can be stained by the basic dye. And later the material which is present inside the nucleus, that material was given the term chromatin by the Fleming. The nucleus was discovered by Robert Brown and chromatin was discovered by Fleming, and chromatin can be stained by the basic dye. Clear? Now see here, beta. Chromatin will condense to form chromosome at the time of cell division. That is why we say that in place of nucleus, you find chromosome. In the cell, in place of nucleus, you find chromosome at the time of cell division. Now, what is chromosome made up of? Chromosome is made up of DNA and protein. Now, which protein? They are made up of histone protein. They are made up of histone proteins. Clear? Now, what is role of nucleolus? Nucleolus is the one which is going to make, ribosomal RNA. Now, what is ribosomal RNA used for? See carefully. Ribosomal RNA, when combined with protein, they form ribosome. They form ribosome. Understand the basic concept. Now, who is making the ribosomal RNA and how the protein? The central dogma of biology says that DNA will make RNA. This process is known as transcription, which can occur in the nucleoplasm only. Inside the nucleus, it can occur. And RNA will be changed into the protein language inside the cytoplasm by the process that is known as translation. So translation will be occurring in the cytoplasm. Transcription is occurring in the nucleoplasm and translation is occurring in the cytoplasm. Now, apply the logic, beta. Please apply the logic. If you have to make chromosome, means you want protein, because chromatin will condense to form chromosome. So it means chromatin is also having protein. So it means logically protein should enter inside or not. But protein can be made only when the RNA is made. So it means RNA should go out or not. So RNA should go out or not. Because when RNA will go out, then only translation will be done, and then only protein formation will be done, and protein will come inside. So nuclear pores are created so that they can maintain the traffic movement of macromolecules. Macro, these protein is polymer of amino acid. RNA is a polymer of nucleotides. So traffic movement of polymer can go on. That is why nuclear pores are created. Is it clear? So RNA has to come out. Now see the NCERT carefully, beta. See. Interphase nucleus, when it is not dividing, means interphase has highly extended and elaborate nucleoprotein. Nucleoprotein means nucleic acid and protein fiber called chromatin. It is having nuclear matrix and it is having one or more spherical bodies called nucleoli. So this is the nucleus contain. Nucleus contain what? It contains nucleolus, it contains chromatin. Okay, number one is nucleolus, number two is chromatin, and it is containing nuclear matrix known as nucleoplasm. And obviously nuclear pores are present. Electron microscope has revealed that nuclear envelope, which consists of two parallel membranes, space between the two membrane is known as perinuclear space 10 to 50 nanometer. So you can learn this way. This is your perinuclear space. This is your perinuclear space nearly 10 to 50 nanometer. And if you check my diagram where nuclear pore are created, the two membranes are united. They form a barrier between the material present inside and in the cytoplasm. Outer membrane is continuous with the endoplasmic reticulum and it will be a ribosome. At a number of places, nuclear envelope is interrupted by minute pore, and these pores are formed by what? Pores are formed by fusion of its two membrane. At the pore, you can't draw it this way only, okay, fine, I'll draw it this way. Okay, no. I'm not drawing it this way. I have to fuse these ends. Is it clear? This fusion end is done this way. These nuclear pores are the passage through which movement of RNA and protein molecule take place. Please, this is very, very important. Through which movement of RNA and protein molecule takes place. Up till here, clear? Now see, I told you nucleoli. Nucleolus is a spherical structure present in the nucleoplasm because nuclear matrix is known as nucleoplasm. And content of nucleus is continuous. Why? Because it is not surrounded by membrane and it is a site for active ribosomal RNA synthesis. And you know, more ribosome means more protein. It means the cell which is working as a gland, cell which is going to work as a gland will be having more nucleoli. So large and more nucleoli are present in the cell which are carrying out protein synthesis. Because glands means enzyme generally mainly they are protein. Is it clear? Now, generally what is going on, beta? There is one nucleus per cell. But we have already studied in biological classification that paramecium is having two nucleus per cell. Normally, there is only one nucleus per cell, normally. But variation in number of can be seen. Who is having more than one nucleus? Paramecium. And some don't have nucleus only. They are living but don't have nucleus. Example, RBC. And sieve tube cells, they are living without nucleus. This much is clear to you. Now, one more thing, try to understand one more concept. Let me rub it here, this area. I hope you have understood, so I'm rubbing it. This much you have understood. Now see, beta. From the outer nuclear membrane, this system is coming out. This system is coming out. What is this system known as? This system is actually an endomembrane system, which is including ER. This is Golgi body. I'm drawing it Golgi body this way. And this is lysosome. Now tell me one thing. Nucleolus is making ribosomal RNA. And when protein are going inside, so I can say that nucleolus is going to send through the nuclear protein subunit of ribosome. subunit of ribosome is coming out. Subunit of ribosome will come out. Please highlight that point. Subunit will come out. Now, beta, I'm using this color for subunit. So I think so when the ribosomes are coming out from the nuclear core, they will be they'll be attaching themselves to the outer membrane or not. And outer membrane is continuous to give rise to ER. And when they're attaching on the ER, they are giving the appearance rough. So we call it as rough ER. So if nuclear membrane is there, obviously it will give rise to rough ER. Understood? So this is the ribosomes which are attached on the outer membrane of the nuclear membrane. Now, if I ask you, this was PMT question, 2 times it was asked, who give rise to rough ER? What will be your answer? Outer membrane nuclear, inner nuclear membrane. Outer nuclear membrane, inner nuclear membrane. Outer nuclear membrane. Now, if suppose I ask you who give rise to nuclear membrane in the telophase, rough ER ki smooth ER? Beta, who give rise to nuclear membrane in the telophase? Do you know in the telophase they make nuclear membrane back or not? So tell me who give rise to nuclear membrane in the telophase? Rough ER ki smooth ER? Rough ER. Your people are muted, you can't give me the answer. This is rough ER. Rough ER will give rise to nuclear membrane. The nuclear membrane is giving rise to rough ER, and the where ribosomes are not attached, that is smooth ER. So rough ER is near to the nuclear membrane, smooth ER is away from the nuclear membrane. Clear? Now see the NCERT carefully. This line. See this line, I'll highlight it. Outer membrane usually remain continuous with the ER and also bear ribosome on it. The outer membrane is continuous with the ER and bear ribosomes. This much is clear. Now I told you when the cell is ready for division, at the time when the cell is ready for division, so during prophase, nuclear membrane, nucleus, everything get disappear. When everything get disappear, what you're finding? You're finding chromosome in place of nucleus. See this line. Line this carefully. Interphase nucleus has loose and indistinct network of nucleoprotein fiber called chromatin, but during cell division, you will find chromosome in place of nucleus. They are framing this way line and the student is getting confused. During cell division, you find chromosome in place of nucleus. Yes, it is true. You find chromosome because nuclear envelope, nucleus, everything disappear. I told you what is chromatin made up of. Chromatin is condensing to become chromosome, which is made up of what? DNA and basic protein, histone protein. Some non-histone protein also, and also RNA. Is it clear? Now let me explain you the diagram. 1 minute. This is chromosome. And when we are seeing the chromosome after interphase in the prophase only we see, so obviously it has come from the S phase, means sister chromatid has been made. Means DNA replication has been done, because you know DNA replication is done in the S phase interphase. On the either side, this is your centromere. First of all, this structure is known as centromere, known as primary constriction. Primary constriction. And these two are your sister chromatid. These are sister chromatid. And on either side of the primary constriction, you're finding this proteinaceous disc-like structure. This is known as kinetochore, where the spindle fiber will attach. Kinetochore, where the spindle fiber will attach. So please make a diagram, it will help you to learn also this way. Kinetochore, where spindle fibers are attached. These are spindle fibers. Clear? Now, depending upon the position of centromere, right now in this diagram, the centromere is exactly in the middle. Both the arms of the chromosomes are equal. But if you see in this diagram, slightly unequal, one big and one little small. See in the next, one very small and one very big. And here it is reached to the tip. So this is known as metacentric, which will appear as V shape. Please note down, this is again PMT question. It will appear as V shape during anaphase. This is submetacentric, which is appearing as L shape. This is acrocentric, which is appearing as J shaped. This is J, okay, J shape. This is telocentric, which is appearing as I shape. So V, L, J, I, depending upon meta, submeta, acro, or telo. And these are, if you see carefully, again, one more question. Here I am saying this is short arm, this is long arm. Again question, long arm is known as Q arm, short arm is known as P arm. My learning tips is keep Q is big, peon is small. P for peon, Q for queen. So short arm is known as P arm and long arm is known as Q arm. Here also we can use this terminology. Here also we can say P and you can say Q. In meta and submeta, we can use this term, sorry, submeta and acro, we can use this terminology, not in the meta, because in meta, arms are equal. Some chromosomes are having some additional constriction. This is additional constriction. So this is your primary constriction. And this is secondary constriction. And because of this secondary constriction, you are finding this structure. And this structure is known as satellite. This is known as satellite. And because of this, this chromosome is known as set chromosome. So please, again, one question. Is secondary constriction known as satellite? Is secondary constriction produce satellite? Secondary constriction produce satellite. It is not known as satellite, please. Clear, beta? You know very well human DNA is 2 meters long, which is distributed in 46 chromosomes, means 23 pairs. Every chromosome has primary constriction known as centromere, on the either side of which is a proteinaceous disc-like structure known as kinetochore. Centromere holds 2 chromatid known as sister chromatid. Now, based on the position, you are classifying them as, this is your metacentric, this is your submetacentric, both are submeta. This is your acro, it is not telo, acrocentric. Long arm, write down it is Q arm, and short arm, write down P arm. So metacentric chromosome has middle centromere, submetic is having little bit shift away from the middle, and acro means situated close to the end. And telo means it has reached to the terminal. So I think so this much is clear to you. Nucleus is clear to you, beta? Any doubt in this? Few chromosomes are having non-staining secondary constriction at a constant location. It gives an appearance of a small fragment known as satellite. Fragment is known as satellite. The secondary constriction is not known as satellite. Remember this. I think so this much concept is clear. Now we can start with the endomembrane system. Now see, beta. I'm just drawing this much nuclear membrane. This is your ER. This is your luminal compartment. Luminal compartment. Luminal compartment. And this is your extraluminal compartment. You know very well, any space bounded by the membrane is known as lumen. So this yellow color is your luminal compartment. And this is your extraluminal compartment. Clear? Now let us see here. Beta. So electron microscope studies of eukaryotic cells reveal the presence of network is also known as reticulum, of tiny tubular structure scattered in the cytoplasm called ER, which divide the intracellular space, means inside the cell cytoplasm area where it is present, into two compartments, luminal compartment, which is inside ER, and extraluminal means it is cytoplasm. The two compartments are there. Now, ribosomes will be attached on it. When the ribosomes are attached, beta, apply the logic. Ribosomes should attach in such a way, this is one subunit, messenger RNA, largest subunit, and the newly formed protein should come out into the lumen compartment or not. Newly formed polypeptide, primary structure, should come in the luminal compartment. If it is coming in the luminal compartment, means pore should be created or not. So ribosomes are attached on the ER with the help of glycoprotein that is known as riboforin 1 and riboforin 2. This is your additional information. I'm just teaching, you can make a note of it. Riboforin 1 and riboforin 2, so that ribosomes stick there, attach there, and newly formed polypeptide, this is your primary structure of protein, is coming from one luminal compartment to second luminal compartment. It will come this way, come here, from here to here, here to here. It is coming, and finally it will be given to the Golgi. It will be given to the Golgi because Golgi is going to do the packing. Golgi will make its secondary structure, tertiary structure, or it will add the glucose to make glycoprotein, whatever it is. Now this piece of ER, if you see this piece of ER, I'm drawing the ribosome. This is known as raphia. This is known as raphia. Clear, beta? So this is your raphia, and this ER is your smooth ER because it does not have ribosome. So logically, when rough ER is having ribosome, it will make protein. But smooth ER will also make something. What it is making? It is making lipid. So I'm using green color for protein. Try to understand. From here, one vesicle is made, and this protein is now fusing with the Golgi. It is going to fuse with the Golgi. Now, smooth ER, I'll use this color, please focus. Smooth ER is making lipid. It is making lipid. So this will be also packed in the vesicles, and this lipid will be given to the Golgi. The same, it will be also given to the Golgi. The Golgi is getting the protein from the rough ER. It is getting the lipid from the smooth ER. Now to this, they can add sugar. Now, if they are adding sugar, you will say they are making glycoprotein. If they are adding sugar to the lipid, we will say glycolipid. So glycoprotein and glycolipid formation is done by the Golgi. But first see the ER. Let us see. ER is showing ribosomes attached on their outer surface. ER bearing ribosomes on their surface is called rough. And in the absence of ribosome, they appear smooth, so called smooth. I think so is easy. Rough ER is frequently observed in the cell, which is actively involved in the protein synthesis and secretion, because rough ER is meant for making protein for secretion, means outside the cell. They will make the protein so that they can be used by the outside the cell, not inside the cell. They are extensively and continuous within the outer membrane of the nucleus. This we already studied. Smooth ER is what? It is the major site for making lipid. And you know very well, all our sex hormones are steroidal hormones, sex hormones only, cholesterol. They are derived from the cholesterol, like we say, your estrogen, progesterone, testosterone. They are steroidal hormone. They are also synthesized in the SCR. Clear, beta? Now, Golgi, for this, you try to understand my diagram first. In the Golgi, do one thing. Right? This phase is known as cis phase, also known as forming phase, also known as convex phase. Now, after this packing, what is going to happen? The Golgi will make tubule, and it will make the vesicle in which the glycoprotein is released. The glycoprotein or glycolipid is released. Glycolipid is released. It is not only coming this side, it can come this one, this vesicle can come to this side also. Okay, it's not it will come only in the first, it can come this side. Now these are released. From this side, the vesicles are released in this form or either this form. These vesicles, if they are remaining in the cell, then these vesicles are known as lysosome. And you know, lysosome is rich in the enzyme protein. So from which side it is released? It is released from this side. You will say trans phase. You will say maturing phase. You will say concave phase. So please learn, this is cis phase, forming phase or convex phase, and opposite is trans phase or maturing phase or concave phase. Now see the NCERT. So see beta, first of all, who has observed? Camilo Golgi has observed reticular structure near nucleus. They are named after him only, we say Golgi bodies. They are flat, disc-like structure, cisternae, this much in diameter. They are stacked parallel. Number can be varied. They are concentrically arranged around the nucleus with distinct convex or cis or forming. Three names, convex, cis or forming, is towards nucleus side. And concave, trans or maturing phase is present towards the cell membrane side. And they both are entirely different, but they are interconnected. So this way you can make once again diagram. They are interconnected. They are connecting them, and cis means forming, means convex. Trans means maturing, means concave. And I told you what they are doing. Their primary function is for packing. Primary function is for packing. Material which is relieved, which they received, okay, which they received to be delivered either inside the cell or outside. Secretion means outside. Material are packed in the form of vesicles, where they, from the ER, which they have to pack, the ER is releasing the material, which fuse with the cis phase, move towards the maturing phase. That is why Golgi is always found associated with the ER. Clear? Number of proteins synthesized on the ribosomes are modified in the cisternae and they are released from the trans. So we say Golgi apparatus is very important for making glycoprotein or glycolipid. Now, if suppose they have made the vesicle which is remaining inside the cell, that will work as a lysosome. So lysosomes are membrane-bound vesicle structure made by the process of packing. That is why these three organelles are the part of the endomembrane system, because their work is interconnected. Isolated lysosome vesicles have been found rich in the enzymes. Which enzyme? Hydrolytic enzymes, which are working at acidic pH. Acidic pH. Hydrolytic enzymes work at acidic pH, and they can digest every biomolecule. In case of the plant cell don't have the lysosome, so they are having the vacuole. So that is why endomembrane system, when we study of the cell, we study ER, Golgi, lysosome, and vacuole, because in the plants, vacuole is taking the function of the lysosomes. The vacuole is also membrane-bound structure present in the cytoplasm. It contains water, sap, excretory product. Please remember excretory product and other material also. It not only contains the food and minerals, but it contains water. It contains the waste also. It is membrane is known as tonoplast. It occupies 90%. Tonoplast is also like a cell membrane, so semi-selective permeable. It allows the transport of number of ions and other material against concentration gradient. It means it is also using active transport method. That is why the concentration in the vacuole is always high as compared to the outside. And because of this, water keeps on coming, osmosis concept, and cell goes for elongation, and the cell turgidity is maintained. In amoeba, contractile vacuole is present, necessary for osmoregulation, and in protista, food vacuole is present. We studied also paramecium, amoeba, they are making food vacuole. So I think so endomembrane system, we see now, we see the heading beta. What does endomembrane system include? See here. Endomembrane system, because their functions are coordinated, the system include ER, Golgi, lysosome, and vacuole. It include all this. It will not include mitochondria, chloroplast, and microbodies because their functions are not independent. Sorry, their functions are not dependent on each other. Their functions are independent. So I hope so. Up till here, your concept is clear. Just go through it. If any doubt is there, then ask.